Thank you so much for being invited to present at this conference, at this PhD course. I must say you have an excellent group of speakers. I'm not counting myself then. I might even add that I'm employed by the Statens Serum Institute as well, and been working at Statens Serum Institute since 2010, helping out to set up the National Biobank facility. So today's topic, my presentation will focus on biobanks and precision medicine, but I think we've heard a lot about that already today. But I might give you a slightly different perspective, or as we say, there are many ways to Rome. I don't know if we will ever reach Rome. I think UK Biobank have gotten there for sure. But I'll focus more on the sort of Norwegian perspective as well. This is from a paper in Nature in 2016 by Schork et al. And it focused on what they called imprecision medicine. And as we probably know from clinical practice, same treatment does not have the same effect on different patients with the same disease. So a systematic approach to identify the right treatment for the right patient at the right time is the only option. Apologies for the spelling errors in the slide. Numbers needed to treat to have a successful effect in one patient varies from 3 to 24. And I think this is quite amazing. So for statin lowering drugs, statins for cholesterol lowering drugs, you need a substantial number of patients to have successful treatment for one. On the other hand, you can experience a number of side effects, and there are also significant costs involved. So we need to do this better. How can biobanks play a central role at the cutting edge of precision medicine? Well, some of the more necessary prerequisites are to have a comprehensive population cohort with state-of-the-art biobank, well-annotated high-quality biological samples, personal identifiers for registered linkage and access to registries for collection of clinical outcome. As Naomi Allen mentioned, it helps a lot to have a prospective longitudinal design with the ability to do repeated measures. You must implement the FAIR principles to be an attractive research resource, meaning your data must be findable, accessible, interoperable, and reusable. You must your aim must be to set up a sustainable cost recovery model and make your biobank attractive to the wider research community and the industry. So in Norway, we have since 2010 been working within a national biobank infrastructure, has been funded by the Norwegian Research Council by close to 250,000 Norwegian kroner over these 10 years. And it involves all the regional health trusts, all the major Norwegian universities with medical faculties, which are also operators of large population-based studies. It includes the National Institute of Public Health and the Cancer Registry of Norway. We have also been part of this European Biobank network, BBMRI ERIC, since 2013 as an observer state and as a full member since 2016. This is a quite messy slide, but as I understand, you'll have the presentation later and can read it more carefully. But I'm trying to illustrate how we can set up an integrated national model for precision medicine based on population studies and clinical application. So in the left-hand panel are listed the major population studies in Norway and their contributions related to genotype data. So in total, we have approximately 400,000 participants that have been genotyped. These are consent-based studies, all population-based. And in addition to genetic data, we have produced an increasing number of other omics data, proteomics, metabolomics, as well as health data, self-reported and measured. Based on funding from the Research Council, we are now working on a national population genomics archive that could be accessible for all researchers. We will provide data, if we can find a good solution for that, to a national health analysis platform, which I suppose could, to some extent, be comparable to the Danish Forskermaskin, which will hold register data from all national registries, clinical data from electronic health records, and also hopefully population-based data. The whole idea by setting up this is to give a researcher one portal for access. There is a already established portal called helsedata.no, or Health Data Service, that you can already find on the web. Then in the right-hand panel, I've listed some of the clinical features or the improved diagnostics opportunities based on clinical translation of data provided by population studies. So some of the clinical tools will, of course, be clinical biobanks, ICD codes, pharmacogenetic studies, randomized controlled trials based on recruitment from population studies, artificial intelligence, machine learning, and improved risk prediction, establish effective timelines for studies of genetic risk and exposure prior to disease, look at disease progression, longitudinal data, repeated measures, trajectories, network analysis. You have some excellent environments in Copenhagen handling this, headed by Søren Brunak. And then we can obviously also improve diagnostic criteria. This has been done, for instance, related to diabetes based on population studies. And there are a number of other stakeholders, industrial collaborations, and also public-private partnerships. So the precision medicine initiative that we are now launching in Biobank Norway is then to establish a national forum for personalized medicine with all stakeholders, coordinate activities and funding of new large-scale genotyping, sequencing, and omics initiatives, including public-private partnerships, identify and establish relevant use cases for personalized medicine within different traits or phenotypes. Based on selected use cases, test the integration of large-scale health and molecular data from population biobanks and population studies with phenotypic information and relevant biomarkers from clinical biobanks and national registry data on the health analysis platform. Establish solutions for data integration and develop standard operating procedures to ensure that use of similar models for other disease categories and phenotypes will be in place, and collaborate closely with LC expertise to address GDPR-related challenges, which are not trivial. So since 2002, I'm being in charge of the Hunt Biobank based on the Hunt study. And in this slide, you can, I present the schematic setup of how the Hunt study has been conducted and the structure of the biobank. So the Hunt study is a population-based study in the county of Trøndelag in central Norway. This initiated in the mid-80s, has been repeated now 4 times, mid-90s, 2006 to 2008, and the latest 2017 to 2019. So I listed a number of participants and attendance rate in this slide. And you can also see there's a substantial overlap between studies. And up until now, there are 20,000 participants that have been through all 4 studies. In Hunt 1, we did not collect biological material, but since 1995, we have genotyped all participants available with available biosamples. And as you know from other Nordic countries, we have some excellent registries. Could be regional, national, where we can retrieve clinical endpoints outcomes by linking data from these prospective screening events till incident disease outcomes later in life. Hunt Biobank have now 100,000 donors. We have DNA from 100,000. We have serum, plasma, urine, RNA, vital frozen cells from a number of participants. We also established a national DNA biobank, collecting a vial of DNA from all major population studies in Norway. In Hunt 4, we also collected saliva, fecal samples, and not only fecal samples from the participants, but also from their pets or dogs in a project called One Health. There's a significant scientific output based on the Hunt study. Now, these are some simple or straightforward presentation of phenotypic data. It's not an overview of COVID-19 cases in Norway. It's the development of obesity from Hunt 1 till Hunt 3. And I can promise you it's even worse in Hunt 4, where about a quarter of the population now are obese or with the BMI above 30. Similar data for both genders. To have a more systematic approach to genetic epidemiology, we have, as I mentioned, genotyped 90,000 Hunt participants using a chip array, the human core exome, with 604,000 genetic markers, including 60,000 custom SNPs based on sequencing of a couple of thousand of our participants. This has been imputed in a traditional way. And we have also involved now more than 60 clinical sub-studies to do more in-depth analysis of how genetic variation affects various phenotypes. This includes more than 150 collaborating clinical researchers. There are, of course, challenging ethics with these, several variants, such as familiar hypercholesterolemia or BRCA mutations. And we have also studies ongoing on systematically return of results to selected participants in collaboration with departments of medical genetics. And this is only possible if we have upfront asked the participants if they want this sort of feedback. So in Hunt 4, we asked participants if they want feedback of results, if the genetic information obtained by result in potential treatment or preventive measures. 93% said yes. And we followed up with asking if they were willing to participate in follow-up studies based on genetic findings with no clear clinical impact, but of course of great research interest. And almost 90% also consented to that. So now we've been doing GWAS analysis on I think, currently up to 2,000 binary or quantitative traits based on our genotype data and ICD codes, and also around 7 to 10,000 unique variables collected from the hunt study itself. In parallel, we run a so-called FIVAS analysis, where we select different SNPs and run them against a number of different phenotypes. So this is just an example of the relationship between an ApoE variant linked to both Alzheimer's disease, blood lipids, and dyslipidemia is probably not very surprising, but if we do this systematically, we might come up with genetic variants that affect traits that's not immediate. So we have discovered rare variants. One example here is a variant representing a protective gene against myocardial infarction. However, this TM6SF2 gene also shown an increased risk of fatty liver disease and an increased risk of type 2 diabetes, so it was not likely to be the best drug target. But I know there are a number of publications on this gene in the recent years. We're part of a larger collaboration with the Broad Institute, with Decode and others, identifying protective genes against type 2 diabetes. And lately we have a study, conducted a study on atrial fibrillation, collecting 60,000 cases and almost 1 million controls. Where the first author is Jonas Bille-Nielsen, an excellent clinician and researcher in Copenhagen, and where we identified genetic variants that had a primary effect on the main effect was on the development of the fetal heart, and not on conductivity of the heart. So now we're looking at the clinical consequences of this finding and whether that might alter anticoagulation strategies, which we want to test out in randomized clinical trials recruiting from well-described population studies. As Naomi pointed out or showed you earlier today, they have had a fantastic collection of data, and I think this is how the biobanks now are developing, trying to convert bio specimens into large-scale data sets. We're trying to get there as well. So we've done some interesting work on within proteomics using the SomaScan from SomaLogic to predict risk for secondary cardiovascular events, which was actually very precise based on only 9 proteins. And now working on metabolites based on this Nightingale platform, and also now negotiating with the Metabolon to use their panels.
Just a short notice that you have 4 more minutes.
Yep. And we hope to then be able to analyze 75,000 participants that have attended to 3 different surveys. I have established a cloud solution. I asked Naomi about this, how they've solved this, and it's not easy. So this is one way of trying to organize our data within GDPR regulations. There is also a precision medicine heart study. It's coordinated by Henning Brungaard at Rigshospitalet in København. Henrik Gullum is also part of this, where we are looking at the opportunity to develop an ischemic heart disease algorithm to be able to tailor the treatment more precisely. Then I said I would show you a few slides on the Nordic Society of Human Genetics and Precision Medicine. I don't have to go through these slides, but they will be in my presentation. And that's what was mentioned earlier today by Mark Daly. So here you have some contact details. You have names of the legal working group and reference to a publication from Nature Genetics last year describing a rollback for precision medicine initiative in the Nordic region. So that's about all I want to say. Thank you very much.
Thank you, Kristian.